Chapter 25, Immunizations and Vaccinations. So we'll go over all the different types of immunity that's involved, what if active immunity and the history and type of vaccines and the advantages and disadvantages to that, and what affects the immunogenicity. Some examples of passive immunity, what it is, its benefits, and its disadvantages, and adoptive immunity. So you should know this by now, hopefully. Immunity is the condition of being resistant to a disease. Immunization is the process by which immunity is acquired, which can be done several ways. So there's 3 different types of immunity, and this is going all the way to the beginning of immunology. There's active immunity, passive immunity, and adoptive immunity. So active immunity occurs after stimulating your own immune system to mount an adaptive immune response. So that means it's highly specific to a particular antigen. So that can be either the cell-mediated or the humoral immunity. So that usually will mean a natural exposure to the infection. You get exposed to what bacteria or whatever virus, you will produce antibodies to it. Now, when it becomes artificial, that's where we have administration of a vaccine. So we are going to develop immunity to a virus after receiving an inoculation of whatever bug it may be. Perhaps measles is a good example to use here. Passive immunity is transfer of antibodies from somebody that has already developed antibodies. So that would be a host that's been immunized to an individual that is not immunized. An example of that is transfer of a mother's antibodies to the fetus or infant. So if this were the fetus, that would be IgG going through the placenta, through the breast milk, that would be IgA. So that would be going to the infant. So there is passive immunotherapy that you can use. It's a commercially prepared antibody preparation. It's where they usually will pull human antibodies from people that have had exposure, and that can be given to people that are immunodeficient. You know, like any kind of antitoxins or anything like that. Winrow or IVIG is another example of type of passive immunotherapy. Adoptive is transferring cells of the immune system. So that would be like a lymphocyte from a host that's been immunized to an individual who has not been immunized. For instance, when we have stem cell transplants, that's a type of adoptive immunotherapy into a patient that's gotten like a lot of radiation to obliterate or ablate their bone marrow, and it's given to them to reseed the bone marrow. So what are the advantages and limitations of that? Active immunity usually will induce a very long-term protection. It takes a little bit. Remember, you got to go through the primary and secondary immune responses. So it takes some time to develop. Passive immunity is just an immediate protection because remember, we're giving ready-made antibodies. So this is temporary. There's no memory produced, where in active immunity we do have memory produced. Now, sometimes passive immunity can cause hypersensitivity, so people can get allergic responses due to this. Adoptive immunity is usually the transfer of cell-mediated type of immunity. So it's when a patient doesn't have their own immune cells, they've been depleted, or they can't produce them. So that's the advantages to it. Some limitations is that possible rejection of these allogenic cells, in other words, coming from the donor to the patient, so we can get a graft versus host disease kind of rejection here. So vaccines are suspensions of antigens derived from a pathogen, which, as you know, is a lot of different things, and that will induce active immunity. So it's considered an immunoprophylactic thing that we give that will prevent disease by giving immunization to a somewhat healthy, competent person. So there's a little history behind it. In ancient China, they actually did some kind of protection against smallpox. And of course, the one that we probably all know about is Edward Jenner using cowpox lesions to vaccinate people against the smallpox. Louis Pasteur actually used what was called attenuated, which means it's a virus or bacteria that's been weakened by either the use of chemicals or growing it at different temperatures or repeated growth in vitro to weaken the pathogenicity, virulence of the actual organism. So some of those vaccinations that were used back in the late 1800s, like rabies, anthrax, cholera, were some of the examples that were done. So the 20th century, which was the last previous one, is considered the golden age of vaccine development. So by the use of attenuating organisms, prevented a huge use of vaccinations for that. We also developed antitoxins. In other words, we have development of toxoids, which is inactivated bacterial toxins like diphtheria. Tetanus is a big one for that. Then we have attenuated viral vaccinations, the MMR, measles, mumps, rubella, varicella, polio, all those attenuated viral vaccines out there. Then we can have production of glycoconjugates, so that's like bacterial polysaccharides that will be linked to a protein to prevent vaccination production. And then they came up with recombinant, and so that would be more of a genetically engineered vaccine. And actually the first one for that was hepatitis B. So the more live stuff that's given for attenuated vaccine is like influenza, herpes zoster, rotavirus that's out there. They have glycoconjugates, multivalent ones that can be used for pneumococcal and meningococcal kinds of vaccinations that are out there. Recombinant vaccines that we have out there right now is for HPV and for shingles. And of course, the development of vaccines is constantly on the change, as we can see from that COVID that's going on. So for a conventional run-of-the-mill type of vaccination that we have out there, so how do we prepare our antigen for this? So you can get the whole organism itself and you can attenuate it. So it's live attenuations, like we said, temperature, growing it multiple times to weaken the virulence of it. Or you can inactivate it by different kinds of chemicals out there, which we'll talk about in a little bit. Or you can use part of an organism. This is considered a subunit vaccine out there. So it's showing you different examples of that. A polysaccharide, which is a type of glycoconjugate. Or we can use purified proteins or recombinant proteins for that. So the attenuated vaccines, like I say, are live, but they are weakened, either viral or bacterial. So, like I said, we grow these organisms under abnormal conditions, so they are not considered pathogenic, but are still capable of eliciting an immune response that we need. So one of the live attenuated vaccines they have for bacterial diseases is BCG, which is used as a Mycobacterium bovis they use to grow, and it protects against tuberculosis in some parts of the world that we have. And there's also for typhoid fever, which is Salmonella typhi, which is actually a mutant strain they will use for that also. Polio is a different serotypes of the polio virus that are out there. It was given orally. Each has its unique antigen, and we can identify all those by serological typing. Influenza, there is a nasal mist that can be given for different strains of influenza A and B out there. Rubeola for measles, rubella for German measles, mumps, and varicella for chicken pox, and then there's one for shingles. Well, shingles is given, later. I think it's 50 years old or more. So what are the advantages and limitations of a live and attenuated vaccine? Is because they will stimulate both branches of the immune system, so both humoral and cell-mediated. It's not recommended to give this to immunocompromised patients generally, but there are reasons sometimes it is given to try to, before they're system is totally immunocompromised, especially like leukemia and lymphoma patients. Before we start giving a lot of chemotherapy and stem cell stuff, we will give vaccinations to try to elicit some kind of immune response out there. Sometimes this can cause interferences with maternal antibodies also. So these attenuated vaccines, they do require certain requirements for handling and storage. As you can see from the COVID vaccine, some of them have to be ultra low temperatures, refrigeration, so it depends on what it is. So inactivated viruses are usually viruses or bacteria that are intact, and they've usually been treated by some kind of chemical or some heat kind of thing to kill it, like the IM for polio, which is a salk vaccination. Influenza is done that way. Hepatitis A is also in that type of an inactivated vaccine that's out there. So they are usually safe to give to immunocompromised individuals. It will still stimulate the humoral immunity, but not really cell-mediated immunity. And usually with these types of vaccinations, 2 or more booster shots are required to give a type of protective immunity. Okay, the subunit vaccines, and they're usually more purified components of a pathogen, like the toxoid stuff, like diphtheria and tetanus, polysaccharides, purified proteins, and recombinant proteins. So they're purified components of a pathogen that are able to stimulate an immune response. So toxoids are the toxins from certain bacteria, are chemically inactivated, so now they won't be considered pathogenic, but still able to stimulate an immune response. So some examples of those would be like diphtheria and tetanus. The polysaccharides are purified polysaccharides from usually bacterial capsules, and examples that we use for that are streptococcal pneumoniae, H. flu, and Neisseria meningitidis. But a lot of those will require a carrier protein to be with it, because it's more of a hapten, it needs a bigger amount so that we induce an IgG production and give long-term type of immunity. Purified proteins are biochemical components of a microorganism. For instance, the whooping cough vaccination, the pertussis vaccination, it's actually got multiple purified proteins from Bordetella. The recombinant proteins are usually genetically modified. They're usually produced from using non-pathogenic bacteria, yeast, or other types of cells. And one of the big examples for that is hepatitis B. Hepatitis B usually comes from a yeast. That's why they actually will ask you that question when you're getting your vaccination if you're allergic to that. So it will be antibodies against the hepatitis B surface antigen. The HPV vaccination is actually made from genes from the L1 protein, and L1 is a type of capsid protein for different types of HPV, so it will protect against possible cervical cancers, anal cancers, other types of genital cancers. So these, cannot be used to produce antigens of other proteins. That's why they use recombinant. So here's showing an example of a recombinant vaccine production for hepatitis B. So here's the hepatitis B virus here. They isolate the surface antigen. Remember the hepatitis B surface antigen? They insert that into a yeast genome. They actually will grow it out to produce more hepatitis B surface antigen. They will then purify it. They get the virus particles to make the vaccine from it. So what are the advantages of a subunit vaccine? It will induce an immune response to a pathogenic component of a particular microorganism. So it's considered very safe because it's avoiding administration of the actual intact organism. So we're using parts of it. That's why it's called a subunit vaccination. There are some limitations with using a subunit vaccination, because it usually will require 2 or more booster doses, so we get enough immunity, high enough titer to protect the person. Sometimes it will require an adjuvant, to increase immunogenicity. If you remember what an adjuvant is something that will increase an immune response to a particle. And it must be multivalent. In other words, it will give a broad immune response. I like the example they used for HPV, where it was the L1. So we're looking at many subunits. So what are the factors that will actually influence immunogenicity? So this is going back to the first chapters we talked about in immunology. Age. So giving vaccine to the youngest individuals at risk for the disease, so long as it's safe and effective for the particular age group. So you can actually look in your textbook on page 505 and 506, and you can see a chart that gives a recommended immunization schedule for people, for adults and children, and you can see the websites there. Like I said, you know, this has gone on a lot of statistical data to try to determine what is the best time for this to be done. Other things that will affect immunogenicity is the immune status. So it depends on the host, or are they immunocompetent? Remember, age is a big thing about immunocompetence. Somebody very, very young, neonatal, just born, they are not able to really elicit an immune response yet. That's why they get passive immunity from their mother. And when you get older, older in age, it will affect it. And also the health. I mean, malnutrition, any other disease process that's possibly going on, stress, a lot of things can affect the immune status of a person that could possibly make them immunocompromised. So the immunocompetence depends on many things. The vaccine composition, is it alive? Is it attenuated? That seems to be the best to produce a better immunogenic response. Subunit vaccines tend to be less compared to a live attenuated vaccine. So that's why we add adjuvants. You heard me talk about that when we talk about subunits. So the adjuvant, like we said, enhances the immune response when it's administered with the actual antigen in the vaccine itself. So that will stimulate a better innate immune response, which will then, once the innate response is instigated, that will release cytokines, which will activate the adaptive immunity. Also, it will help with antigen delivery system, so it enhances the uptake of the antigen by the antigen-presenting cells that are out there. So, for instance, the dendritic cells. It will activate dendritic cells to help present those to the T cells, which will then get further on into activating, getting the B cells there. So, some examples of adjuvants that we have out there is aluminum salts, oil and water emulsions, Fruin's complete and incomplete adjuvant out there, and you can see other ones listed there. Virosomes. A CpG is an oligodeoxynucleotide that can be used to help bind to the T cell receptors to get a better immune response. So this will result in a faster and a longer lasting response. So it eventually will increase the antibody titer, because you want to try to make sure we get the best immune response possible. That's why it's constantly monitored. Do we need to give a booster or not? So we can follow the antibody titer, be it IgG or IgM. So with this adjuvants, that will help with the cell-mediated immunity, which will then go into being more of the humoral immunity. So depending on what the antibody titers are, will depend on, that will tell us the amount of dosage of antigen that's needed, how many inoculations are required, boosters, timing, repeat boosters, that kind of stuff will determine what we need to do. So there are a lot of different challenges out there right now in the future as to vaccine development. Right now they do have one for Plasmodium falciparum that's being done in endemic areas for malaria. Hepatitis C is still working on, so is TB, but malaria is definitely a very positive thing for us to have. So there's all kinds of new vaccine designs out there. COVID is one of the biggest ones that we've seen with an mRNA type of vaccine delivery. So different types of adjuvants, so there's all types of research that's done out there. So it can be used not in addition to just, epidemic kind of situations, but also is being done for cancer kinds of stuff. HPV is an example of one, talking about like cervical cancer, but there's are different ones that they're coming up with that possibly they can be used to treat cancers, like the one that is a PD-1 blocker, which we'll talk about here in a second. So all these types of vaccinations, this is where we're talking about molecular genetics, the different types of structural biology out there, using bioinformatics to figure out what we need and how to determine what type of vaccine needs to be done. And then we have to be able to monitor the immune response to see what's working and what's not working. So vaccines are very beneficial. They've definitely made a huge impact on the health of humans because it's reduced or eliminated many serious, potentially fatal diseases out there. I mean, smallpox, polio, measles, mumps, rubella, all that kind of stuff. So it's actually decreased morbidity and mortality. It's done a huge, huge thing for that. I mean, they're still in development for like RSV, Epstein-Barr virus, CMV. Still looking for a lot of different types of possible vaccinations out there. And with messenger RNA, the mRNA molecules that actually code for vaccine antigens, they've sparked a huge new interest in generating immunity in a more safe and effective manner out there. So they can be produced rapidly, and they're very effective and pretty inexpensive in the grand scheme of things. And that's what the COVID-2 or the COVID-19 that we're dealing with now. So what we're after with a lot of benefits of vaccinations is herd immunity. So what we're talking about is being able to protect people if not everybody gets vaccinated, but if enough gets vaccinated, that will protect the majority of the people. So here is a picture, and if you look at it in your textbook with 509, you can see this picture a little bit better. So the people in blue are the non-immunized but still healthy. The green are the immunized and healthy, and the red are the not immunized, sick, and contagious. So if you look at the first one up here, no one is immunized up here. So, and you can see the two non-immunized, sick, contagious, and you can see what happens in time. Because of not having immunization, the majority of the people will end up becoming contagious and spread the disease through the population. So if we look at the second one, here's some of the population gets immunized. We still have the same two non-immunized sick individuals that are contagious. And you can see how it does spread, but not as much as we saw in the first one. So the contagious disease will spread through some of the population. Now, if we look at the bottom one, majority of the population is immunized. Still have the two that are not immunized, sick, and then contagious. So the spread of the disease is somewhat contained compared to what we saw in the top slide, top picture. So there are some adverse effects of vaccinations. Because of course, the most common local reaction is the injection site, swells, gets tender, hurts. Some generalized reactions that you get is low-grade fever and ache, tired. Severe reactions are pretty rare because they do a lot of clinical trials to make sure we don't have this kind of problem. Of course, there's exceptions to every rule, so you have to be careful with that. That's why, when you get a vaccination, they want to observe for at least 15 minutes to make sure there's no anaphylactic response. So you can have allergic reactions, which could be type 1 or type 3 hypersensitivity. And then if the vaccine doesn't work, you can get development of the disease. I mean, some people that are immunocompromised, if they get a live attenuated vaccine, it can, it's a small percentage, but it can cause the development of the disease in those individuals. Passive immunity is where we transfer preformed antibodies to a host that is not immunized. Now, this can happen through injection or IV injection, through administration of like the IVIG that we have been talking about, or it can be naturally, like we said, through breast milk or through placental transfer. It can happen that way. But remember, passive does not last. There is no memory developed through that. So here's showing you the example of the passive with the mother. So here's the IgG, placental transfer to the infant. Again, like I said, it only lasts for a few months. Or we can get IgE transfer through the breast milk that way. Again, does not last long. It's a type of passive immunity. Then here's that window that we were talking about, the IVIG, standard human immune serum globulins. We can actually have antigen-specific immune globulins out there. I mean, they actually have one for tetanus. We have one for like rattlesnake, all that kind of stuff. You can have all kinds of immune globulins. That's when you hear about the antitoxin stuff. And then there's monoclonal antibodies out there, which is, you know, the great thing that we have out there. So the IVIG, which also called the standard human immune serum globulin, is a type of gamma globulin, and it's made from a whole pooled amount of serum from thousands of donors. You know, contain antibodies to multiple antigens out there. So it can be used prophylactically to patients that are immunodeficient. There are antigen-specific immune globulins, also can be called hyperimmune globulins. Again, that's pooled serum from donors with immunity to a specific or particular pathogen. Like they do have stuff for hepatitis A and B. They do have for rabies and for tetanus. So we use those to treat people that are unimmunized or people that are possibly exposed to a pathogen. I mean, humans do not get rabies vaccinations, but if they are bitten by, say, a rabid individual, they will be given, you know, this immune globulin that will protect them against this. Multiple series of shots that can be done. Animal globulins are used to prepare from horse serum, equine, base. So they're antitoxins, and it's to multiple bacterial toxins. They use it a lot of times for tetanus, botulism, and diphtheria, like anti-snake venoms. You know, there's a whole bunch of different types of anti-venoms that they have out there. So what they do is these antibodies will neutralize a toxin and or venom that will prevent harm to the host. Because some of the snake venom, spider bites, that kind of stuff, I mean, very, you know, hemorrhagic, neurotoxic kind of events that can happen from those. So this is monoclonal antibodies. Remember, they are made from a single clone of a B cell, not multiple clones, but a single clone, hence the monoclonal. If it was multiple clones, then it would be a polyclonal. So they're directed against a particular epitope of an antigen, and we use those to treat all kinds of things. I mean, if you look in your book on page 513, table 25-2, you will see a whole list that is used to treat cancers and all kinds of autoimmune diseases out there. You know, these are ones you see on TV all the time. And you can tell from the name, the last 4, 5, 6 letters of the name that will tell you where it's derived from. If it's a human or humanized or a chimeric type of base or mouse base. So it can tell you how the monoclonal antibody was actually produced. So, benefits of passive immunization for these monoclonal antibodies is it provides immediate immunity. So, say a person got, you know, stepped on the rusty nail and they got a puncture wound, possibly contaminated from soil, and they hadn't gotten their tetanus vaccination like they should have. We can give them this monoclonal antibodies or this antitoxin to prevent them immediately. But it's a passive immunity. So it does not protect them forever, just for a short period of time. That's why it's important to give vaccinations so we have more of a active immunity there that will last longer and will have memory. So passive immunity can also be used in people that are immunosuppressive. For instance, you'll hear me talk about this. We talked about it once. We're going to talk about it a lot more in blood banking. but a Rh negative mother who gave birth to an Rh positive fetus. There's a picture somewhere in your book. Anyway, so the Rh negative mother who had an Rh positive fetus, what had happened is during birth, usually there's a fetal maternal hemorrhage, and the Rh positive cells got into the Rh negative mother. Because it is foreign, the mother develops antibodies against the Rh positive cells. Now, the antibodies that the Rh negative mother has produced are IgG in nature. So, on the next child in utero, what will happen is because these Rh negative mother has now got IgG antibodies to Rh positive cells, they can cross the placenta and will cause possibly hemolytic disease of the fetus and or newborn. So that will happen with future births. So what they have done is we actually give mothers that are Rh negative, we give them what's called RhoGAM, which is IgG antibodies to the Rh positive. And what it will do is if any Rh positive cells from the fetus get into the mother, these antibodies, this passive immunity, these antibodies will then cover these Rh positive cells because of the antibodies, and it will sort of like cover them up so that will not be considered foreign, and the Rh negative mother will not produce antibodies against it. So that's why it is given. So it's usually given midterm, and it's usually given within 72 hours of delivery. Depending on delivery, then we find out if the baby is Rh positive or negative. So that has prevented a lot of hemolytic disease of the fetus and newborn. So passive immunity, like we said, is very short-lived. We can't develop memory, and you can see 23 days is the actual half-life of IgG. But passive immunity can also induce hypersensitivities, especially to like animal sera. And we talked about using equine sera here. So that would be like a type 1 or possibly type 3 hypersensitivity that can happen. Adoptive immunity is where we actually transfer the cells of the immune system into the patient. And it is performed to increase a lot of times cell-mediated immunity. For instance, in treating cancer patients, we can give tumor-infiltrating lymphocytes that have been activated in vitro. So what they do is they get the patient, we will get T cells out of them. The T cells will then be exposed to interleukins, which will activate them in vitro to whatever particular virus or cancer that we're after. And once they are heightened, we'll say, they will be ready to activate against these tumor cells. They will be reinfused into the patient. So this is called CAR cell therapy, C-A-R-T cell therapy out there. So that's a type of treatment that can be done. Also, transplantation of stem cells into patients that are immunodeficient, so they can try to get the production cells, lymphocytes, restarted. So that's just some basic things of immunity and vaccinations. Just to summarize, remember, active immunity is where the patient will develop their immune response because they've been exposed to the antigen. And usually with active immunity, we do have memory that we can remember this and produce antibodies when re-exposed. Usually will last for a prolonged period of time. And usually active immunity can happen by just natural infection or administering vaccinations. Passive immunity usually involves transferring of antibodies from a host that has been immunized to a patient, an individual that doesn't have antibodies, a non-immune person. So some of the examples that we talked about of passive immunity was talking about the transfer of the mother's antibodies to the fetus. Remember the IgG crossing the placenta and the IgA in the breast milk. So we can transfer immunity from the mother to the fetus or infant, or we can get commercially prepared antibody preparations. Active immunity takes time, but it, like we said, does give a long-term memory because we have memory cells, B cells, that remember how to produce this. That's that secondary immune response that can happen extremely quick. Passive immunity is immediate because antibodies are already made, but it is short-lived. Remember, IgG has a half-life of like 23 days. Adoptive immunity involves transfer of cells from a host that has been immunized to a non-immunized individual, like stem cell transplants or the CAR cell type of therapy, the T cells that have been activated. So you can look in your textbook, in the summary, and in the study guide, and look at the review questions. But immunizations and vaccines is our last chapter, and there's a lot of information that we've covered in this particular chapter, but a very, very important chapter. If you have any questions, you can email, call, text, whatever, and I'll see you in lab.